Hi, everyone. As Jen mentioned, my name is Naheed Kurji, and I have the privilege of both being the CEO of Cyclica and one of the co-founders and the current chair of the Alliance for Artificial Intelligence in Healthcare, the AAIH. So welcome everyone to the next installment in our ongoing series M2M, as Jen just mentioned. I appreciate you all spending the next hour with us. I also want to thank Jennifer and her marketing team at Cyclica for continually putting on these events and engaging the community in what we hope to be an enlightening, informative, and captivating series of discussions. Thank you also to the AAIH for sponsoring the past few and the next couple M2M events. For those of you who are interested, the AAIH has published a few papers recently on its website, which we will add to the chat. One published in June 2022 titled The Life Cycle of an AI System in Healthcare, and another from September 2019 titled A Primer on AI in Healthcare, which was co-published by one of my dear friends and one of our panelists, Brandon Allgood, Chief AI Officer at Valo. And so with that, I think that's a natural segue to go into introductions of the speakers. So Brandon, if you can take it away, please.
Yeah, so Brandon Allgood right now, my position is Chief AI Officer at Valo Health. I've spent about, I don't know, I lose track, close to 20 years in the space of applying machine learning to healthcare. Helped co-found the Alliance for AI in Healthcare with Naheed. I've had a number of startup companies. Yeah, Valo, we are bringing, what I would say is the first digitally native end-to-end pharmaceutical company to the market, and largely based on human data. You'll hear a little bit more about that in the panel today. So thanks a lot for having me.
Thanks, Brandon. And Andreas, if we can move to you, please.
Sure. I'm Andreas Bender. I'm a group leader in Cambridge University. So we analyze data of very different types for 15 years now: chemical data, gene expression data, imaging data. So I'm always quite curious to see where's the signal in the data for safety or efficacy related endpoint. Yeah? Because that's what we do in drug discovery. It's not really about finding ligands, right? That's only means to an end. The question is always, how do you move to in vivo, and where do you have the right data to do that? And so we started a couple of companies for that as well: Helix, Farminable, more recently Pangaea Botanica. Yeah, and that's actually the question I'm always interested in, right? Where's the signal in the data that we deal with? Good meeting, everyone.
Andreas, thank you so much. Looking forward to your comments. And Ali, up next.
Thanks, Naheed. Hello, everyone. I'm Ali Madani, Director of Machine Learning at Cyclica. I'm also currently editor of Artificial Intelligence in Cancer Diagnosis and Therapy, a special topic at MDPI. So I'm leader of machine learning front at Cyclica, and what we do is just machine learning based and AI based drug discovery. And my passion, what we've been doing at Cyclica, is to build scalable technology and rely on combination of expertise that we have to deliver project specific and target specific good drugs and compounds that can get into clinical trials and eventually FDA approval. So, and I'm very excited for this panel.
Ali, thanks. And last but not least, Pat Walters.
Yeah, hi, everyone. I'm Pat Walters. I'm Chief Data Officer at Relay Therapeutics in Cambridge, Massachusetts. A lot of my group's work sits at the interface of computation and experiment. I've been working in the pharmaceutical industry for 27 years. Before that, I got my PhD doing machine learning when it wasn't a fashionable thing to do.
Pat, thank you so much. And for everybody else who's attending this panel discussion, I've had the privilege of sitting on panels or listening to the speakers in the audience. I'm excited for this. I think there's a lot to be learned. I want to give a quick shout out and congratulations to Pat. I think just yesterday or early this week, the American Chemical Society Division of Chemical Information announced that Pat was selected to receive the 2023 Herman Skolnick Award for his contribution in this field, specifically in chemical information. So Pat, congratulations on that.
Thank you.
So jumping into the topic today, we've broken it up into a few themes. First one's going to be on data quality, relevance, and bias. We hear a lot about the importance of data quality in the AI space. We know the adage, garbage in leads to garbage out. But let's go a bit deeper on this to help us understand the implications of data quality in AI. Why is it so important that we get this right? And what are some of the consequences of getting it wrong? I'll turn it over to Ali if you want to kick it off.
Sure. So I want to touch a little bit about the whole drug discovery pipeline. So we start from patients or biological matters, come up with targets going through hit identification, optimization, preclinical, and then clinical. At the end of the day, the ultimate goal is patients to bring drugs into clinical setting. But how machine learning modeling works is to come up with models specific to each sub-problem in this whole pipeline. And then for each one of those sub-problems, what we need to take care of is to make sure we have high quality data and preferably in high quantity. And also we have to make sure about some of the biases that may exist. And sometimes people just, when we talk about biases, they just care about just biases in the society, like race, gender, and so that's true in clinical level, but we have also in preclinical setting and in the drug discovery setting, some other biases, for example, in chemical series that we can talk about in this panel later on in a few minutes. But the processing of data is a major player in coming up with high quality machine learning models, because the algorithms, there are a ton of algorithms out there. And from computational perspective and the hardware perspective, there has been a ton of growth. And it is now the data that plays a major role in coming up with reliable models. And I want to actually give it to other panelists to talk further about this matter.
Yeah, so Ali, as I mentioned in my introduction, one of the things, at the end of the day, what we're trying to do is develop therapies for patients, for human patients, not for rats, not for model animals, right? And so one of the things that is very important is that while yes, we can build machine learning models on mouse data, we can build machine learning models, but we're literally building models of humans. And what we really need to strive to do in terms of data quality is think about human relevant systems. I think biology, experimental design, technology has moved forward to the point at this point where you can, in many ways, there's no excuse not to use a more human relevant system. While yes, commissioning that mouse study at a CRO is cheap and easy to do, but is it the right thing to do? Is that the right data to build your models on? Right. So, more recently, Valo acquired Terra Biosystems for the exact reason that they've developed an amazing human-derived, iPSC-derived cardiomyocyte model, which recapitulates a lot of the functionality of the human heart. Using models like that and that advanced technology, then taking that data, building machine learning models on that, and actually being able to predict clinical outcomes like left ventricular ejection fraction is the kind of thing that we should be thinking about. On the other end, also recently we announced our partnership with Maccabi in Israel. They have some of the highest quality human-based data, longitudinal data. They've been collecting data on 8 million patients for over 15 years, very high quality data. So I think we need to think about not only where does the data come from, but how is it collected, for what purpose, and how we plan to use that. And as you said, Ali, we're in an industry where it's death by 1,000 cuts, and each of those cuts is often independent and needs an independent source of data. And we need to be thinking about the quality of each of those as we optimize compounds and other types of therapies.
And I think that's a good segue to conversations that Andreas, you and I have had over the years, which is, translation into preclinical environments to get to the clinic. And in your introductory remarks, you said it's not just about ligand target binding, but the translation of that. So talk about that. What are the challenges in collating some of that data?
Yeah, that's right. I mean, we heard the term high quality data a few times already, right? And to data, there's many aspects, right? I mean, high quality can mean it's reproducible, right? You have technical, biological replicates and so on. But what I think matters most is actually, and Brandon alluded to that already, predictivity for the in vivo situation, right? If you think about data, and I talk about preclinical data right now in pharmaceutical companies, right? I mean, much of that data, where does that come from? I mean, since the 80s, we went into the area of high throughput screening, right? So what does that mean? We have a large number of compounds to profile in simple assays, right? Not only primary assays in HTS, but what does that mean? You have 10,000 HTS hits, you need to profile those compounds further. So what do? Other high throughput measurements, right? So whether it's solubility or log D measurements or other profiling, let's say, cellular toxicity or BSEP inhibition, or simple assays that are meant to tell you something for the in vivo outcome, right? So BSEP inhibition for DILI, for example, right? And the need was that there were lots of compounds that needed to be profiled. As a result, I think that's still the hangover from HTS times, basically, right? And very simple assays. But now the question is, of course, what's the predictivity of those types of data for the in vivo situation, right? Because that data, and Brendan mentioned the models of models, right, of patients, right? So in silico models, so that's the way I interpret that, in silico models of proxy assays, right, of patients, yeah. And quite often we have that type of proxy data, right? And the question is, does that tell me enough about the in vivo situation? Yeah. And some of the trends are actually quite encouraging, I think. If you go into organoids, for example, yeah, microphysiological systems, yeah, there's still some work to be done, standardization, reporting, and so on. There's still some work that needs to be done, but I think that moves exactly into the right direction, so that we have data that's predictive for the in vivo situation. That's the most important aspect, I think. Yeah, that comes before even sampling or noise and so on. Yeah.
Pat, do you want to add any comments to this?
Yeah, I think it's great that we're talking about looking at things from an in vivo standpoint, but I think there's also another aspect to relevance, which is how relevant is the training data that I have to what I want to predict. So, just as to create an analogy, let's say that I'm trying to build a machine learning model that can identify birds. And if I've trained this model on parrots, and then I present it with penguins, I'm not sure that my model is going to be accurate. And I think we have the same sort of situation in the small molecule world, where I've got literature data, perhaps on hundreds of compounds. But if those compounds look nothing like the molecules that I'm trying to predict, it may have limited value. So I think relevance really spans the spectrum from the beginning with in vitro assays all the way through the relevance of your in vitro assays to a human system.
Excellent. Thank you so much for those comments. And I don't know if there's anything that Brandon, Andreas, Ali, you want to add to that before we move on to the next conversation, which is on the different computational methods. So before switching gears, any additional comments or you want to address, challenge anybody?
I just want to add something. So when we talk about quality or relevance, so it's not black and white. So in reality, it's more of a shades of gray. So, and it's just a matter of how exactly we're defining high quality and just how we deal with this threshold and trade-off of quality versus quantity. So in the case that Pat said, it was a great example. So when we have penguin or just images of different animals, it is clear. So unfortunately, in our area, is not that clear because we are dealing with the statistical similarities, statistical confidences. So one example that we tried to deal with that issue was a transfer learning-based approach that we developed 2 years ago to learn from the quantity of data and benefit from the high quantity of the data that we have. And in a stepwise manner, learn actually from high quality to just balance both benefit from quantity and quality. So that's something important to keep in mind if people in the audience want to actually deal with these kind of situations.
Yeah, one last comment, right? Is I think we've been talking, we've talked about relevance and a number of times, but really we need to think about bias, in particular when we're dealing with human data, right? Bias, I think, is really relevant. It's relevant in all aspects of machine learning, but I would say, when we're getting into clinical trials, when we're doing disease modeling, we need to really be looking at the data and understanding those biases just just as my panelists point out, right, the basic tenet behind machine learning is that the training set is pulled from the same distribution as the test set, right? But in our space, that's like almost never true, right? And so you have to know that you're extrapolating, you have to know the relevance of that extrapolation. And you need to think about bias in one way, again, and I'm a big advocate of this, one way to ensure, at least when you're dealing with human data, thinking about biases by having a very diverse data science team that has many different backgrounds and experiences in life and can help you understand certain hidden biases in your human data. I've had a number of examples where, you know, everything looked great, but because I had a different life experience than one of my team members, they saw a bias in the data set that I didn't. And so by having a diverse group of data scientists in your company, you will end up with a much better, much more relevant models.
Yeah, that applies also to academic groups. I always try to have a diverse composition of group members in the team as well, exactly due to those reasons as well.
Yeah, thanks for that. I think that's one of the biggest topics, not just in the clinical space, but also in the discovery space. Both are really important. And, you know, in the discovery space, Ali mentioned a couple of forms of bias that models or companies that are building these models, deploying these models need to guard against. One is compound series bias. One is negative decoy bias. I think we've seen limitations in AI, ML, deep learning technologies in the drug discovery space, particularly as it relates to kind of the chemistry that have fallen victim to that form of bias. And then obviously in the clinical setting, you know, guarding against homogeneous forms of data is really important. Cyclica, about, I'd say 6 weeks ago, and this is a bit of a shameless plug, we invited Anjali Muller from Roche, who's also the vice chair at the AIH, Rory Kelleher, head of business development for life sciences at NVIDIA, Thomas Clozel from Aukin, CEO of Aukin, and Shweta Maniar, where we had a M2M series on cloud computing for AI and drug discovery and how we can use new methods, new technologies to guard against various forms of bias. And the very next M2M episode that Jen alluded to is specifically on forms of bias in drug discovery and development. And in the chat here, I've placed a link to register for that next conversation. And Brandon, thank you for emphasizing how important it is. I hope you all get to attend. So switching gears now to just generally computational approaches in drug discovery. This may be a bit crass and overly simplistic, but companies in the computational and drug discovery space generally fall into two categories: physics-first companies, structure-based companies, and AI-first companies, knowledge database-first companies. At the intersection are some really interesting approaches that combine both structure and AI. Let's talk about that and what the opportunity looks like, both separately as well as at the intersection. And Pat, given your rich background, as well as Andreas and the others, I'll pass it to you to take it away.
Yeah, thanks. This is, to me, one of the most exciting areas for application of machine learning. We've got a lot of computationally expensive, accurate calculations. So we have free energy calculations, we have quantum chemical calculations. Because of the computational expense, we typically can't run these on large data sets. But one of the really exciting advances of machine learning has been the ability to build models to estimate some of these. So build models to replicate the results of free energy calculations. And by using active learning, rather than being able to evaluate a data set of 100 compounds, we can now evaluate data sets of 10,000 compounds. With some of the work for machine learning for quantum chemical potentials, calculations that used to take a couple of days, we can now complete in microseconds. So this really gives us the ability to calculate things on a much larger scale. And by taking this and combining this with what I think one of the other revolutions in our industry is the availability of synthesis on demand. So now through companies like Enamine, we have the ability to access libraries of billions of compounds that we can get very rapidly and inexpensively. So these combinations of machine learning with physics-based methods and the ability to do rapid synthesis and get molecules cheaply and quickly allow us to test a lot more hypotheses, and I think move programs forward more quickly.
That's a really good point. Thank you so much. I have one comment to make, but I'll pass it to Andreas and Brandon, Ali, if there's anything that you'd like to add.
Yeah, what Pat emphasized, right? That's the physics end, let's say, of drug discovery, and that can certainly speed up things when it comes to hit identification, optimization, and so on, right? To increase the speed in those cycles. Of course, at the other end, you also have the biology angle. And I'm originally a chemist, but I did my postdoc at Novartis, and I analyzed high content screening data. And it was a painful 6 months, and I thought, I don't get that type of data. And that's where I learned how complex biological data can be. And I just realized that's the way biology is. There was nothing wrong the way I did it. That's just the way it is. Yeah. And that's where I realized we also need to take care of how do we translate that forward, right? And with biological data, there's always the question of conditionality of data, right? So what's the dose, what's the endotype, what's the genotype, and so on. That can always impact your downstream translation as well, right? And this is what some of the AI companies can do, of course, looking into electronic health records, right, for example. That's the question, how do you take the biology into account, right? So physics can help you. Biology is very important, I think, to consider as well for the translation later on.
Yeah, I think both my panelists have hit the nail on the head, right? Guilty as charged. I'm a physicist by training in graduate school, and when I approach any of these problems, thinking about choosing the right, for example, choosing the right representation. Choosing the right representation of a molecule, choosing the right representation to build a machine learning model on. I always go back to, what's physically happening? What is the physics behind what is happening? And is there a way to develop a set of features, do some feature engineering that more relevantly reflects what's happening physically? And then I could show many different experiments where if you go and approach it that direction versus maybe what's the most common way to do it in the literature or what most people do, you can show that you get more bang for the buck if you sit down and you think about physically, what does diffusion across the lipid bilayer look like? What's relevant to that? What about binding to a ligand? That's to first order, that's a shape and electrostatic interaction. If you think about those kinds of things, you end up ultimately coming to much better, much more predictive, much more generalizable models. And so I would just say, think about it when you're doing feature engineering.
Yeah, I think all that's very valid points. I want to come back to a comment that when Pat started answering the question, it made me reflect on 9 years ago when we kicked off Cyclica. Our entire thesis was to explore the proteome for a small molecule to identify on targets, to optimize for that, avoid off targets. But we brought a physics-based, structure-based approach to this. And specifically, we built a docking-based algorithm to take a molecule and take a panoramic view of the proteome and go find what it's doing. And what we found was it was just limited. Docking has its strengths, but it also has strong limitations. Predictive power, there was a lot of noise in what we were finding. You're going across, at that time, close to 10,000 different human proteins, a lot of noise. The throughput was limited, it was costly. It just wasn't inspiring. The problem we continued to find was an interesting problem to solve. The approach using just a physics-based strategy was not. And then we looked at just the AI side, and we saw some drastic limitations of just applying kind of data science or ML strategies, because there's only so many targets for which there's high quality quantum data around. But the intersection of combining like strengths and structure physics as well as an AI, I think, is a really cool way. The problem that we want to solve is, let's go explore the proteome, kind of match the chemical universe to the protein universe. I think living at the intersection is important, and it comes back to a central thesis that I think we'll talk on in the next few questions, which is AI is not the silver bullet. We need to see its strengths, combine it with the appropriate techniques, both computational and empirical, to help move something from a target to a clinically viable candidate. And so let's switch gears for a second. And I see a bunch of questions coming through the chat. Samir and Julie see a bunch of questions. I see them fitting topics that are coming up. So please be patient and we'll get to those questions. I want to just build off my comment with a question to Brandon, Ali, whomever else, Pat, Andreas. By most approximates, there are around 20,000 proteins in the human proteome. A vast majority of these proteins, approximately 90–95%, are considered undrugged. Only a small minority are targets for which there is an ample amount of data, whether that be protein structure data, proteins with a lot of known chemical equity, etc. How do you expand the domain of applicability of physics-based and AI models to the undrugged proteome?
Thanks, Naheed. I can have to take the first part of that, right? So I think we touched a little bit in the last one, and you already alluded to it in your question, right? Is what we've found works, and what we found is really helpful. Is again, I don't want to rehash this, but is to think about what's physically happening and develop those representations that reflect the physics. Also remember and think about what we talked about earlier, in that almost undoubtedly you're going to be extrapolating, whether you have whether you're training based on a natural ligand or a set of natural ligands, and then you want to move to something that looks more xenobiotic, something that is more relevant as a compound or as a medicine. You need to think about that extrapolation, employ, as Pat was pointing out, active learning and thinking, extrapolate a little bit, make some compounds, get some training sets, and actually think of more about not just a model, but that modeling loop, and how you speed up that modeling loop, how you develop and collect relevant information in that modeling loop. I am, however, pretty excited about the low data space in many of these kind of more advanced deep learning models, one-shot learning and zero-shot learning models. We've had a number of successes there as a way to start to begin that cycle, because many cases, as you point out, Naheed, some of these don't have any compounds, any even natural ligands against them, especially when you're looking at disrupting things like protein-protein interactions. And so there, I think you need to rely. You have very little to rely on from machine learning perspective, because machine learning is not magic, it's math. As I often am fond of saying, right? You need some data to start. And so going back to the physics, looking at what's happening in that protein-protein interaction, for example, and developing at least one or two compounds, even if they're, practically to the point where they're insoluble because they're, maybe their activity is 50 micromolar, but, and then starting with that one set of data, pulling in a more generalized machine learning model, developing that transfer learning stuff that Ali talked about, and then going for a one shot and building that active learning process. I think we've literally touched on already today all of the aspects you need to think about in a low data environment. It's just bringing all of those things together and thinking about all of them at once.
Yeah, I can add to that. So that was a great point, Brandon. So in total, if you want to talk about it, is mainly a matter of data efficiency. So we have data and we want to benefit the most about for a specific objective we have had. So it could be about representation learning and self-supervised learning to identify representation that could help us in supervised learning setting. It could be a matter of active learning to go for minimum cost and benefiting the most from the money we are putting into generating new data. But at the same time, one thing which is actually the beauty of machine learning is generalizability for the identified patterns in the data space. So one way that we approached it was matchmaking, which was actually successful, to say that we have the whole proteome and we have similarities of patterns between proteins and similarities of patterns between ligands. And then how matchmaker works is it identifies and learns patterns of chemical compounds, proteins, and the binding sites. And then it generalizes using the information we have in the high data proteins and low data proteins to both parts of the space. Then that helps us actually to develop compounds for low data targets. One example was DCAF1, which was a very successful and very exciting project that we had with the Structured Genome Consortium. And we actually deposited the first protein, the first structure of DCAF1 with co-crystal with a small molecule. And that was actually based on this benefit of building a machine learning model on whole proteome and benefiting from similarities of patterns between proteins. And another aspect of it, which was also very important, was AlphaFold, which is tricky. So it has its own pros and cons for sure. It's not definitely something perfect. But if we want to generalize and learn from different proteins, we had to have access to the structures of those proteins. So for that reason, we integrated AlphaFold, and that was also very helpful. But definitely going back to quality and relevance and biases, we had to build pipelines and processes to make sure about the reliability of the predicted structure, reliability of the identified binding sites or so.
Yeah, if I could just build on that a little bit. I think we're seeing a fundamental change in the way that we approach science. Up until recently, I think science was primarily driven by people staring at things and saying, oh, I saw that. This must be interesting. So, back to Andreas's early example of, looking at images from cellular phenotypes. I did the same thing where you would sit down with a biologist and the biologist would stare at the image and say, oh, that's important, and that's important, and that's important. So, write an algorithm to find those three features. I don't think we do that anymore. We just let the machine learning algorithm figure it out. Naheed, to your earlier point about docking, we sort of started out by hand tuning scoring functions for docking and saying, oh, this is important, this is important. Why not just say, here's thousands of crystal structures, figure out what's important. And I think, to Ali's point, now AlphaFold is the primary example of that. We're saying, rather than me looking at protein structures and saying, this is why they fold, we're just going to look at a lot of structures. We're going to acquire that data. Then I think that allows us to start thinking about problems in a very different fashion. So to me, it's now trying to use machine learning to give us insights, use those insights then to drive further machine learning.
Thanks for all that. I'm going to switch gears for a second. I'm going to break from my Q&A, and the next set of conversations for everybody's preparation is going to be on like chemical viability. But I'm going to break from that for a second and go to the online questions. And Samir, I know you're the first one that came through. I'm going to include that in the next set of questions. So I'm going to go first to Julie. Julie asked the following question, and I think I'm going to turn this to Brandon, given that you were talking about the work that you're doing at Valo Health. So given the discussion on using quote unquote high quality data to train and predict for quote unquote drugs versus predicting quote unquote hits, then how would you propose we access publicly available clinical trial data in a machine readable form? Example, in a spreadsheet, data frame with mapped targets, disease, and results easily tracked. Are there any efforts globally to standardize the distribution? Same question for patients. So maybe this is an interesting way to talk about federated learning, some of the work that we're talking about at the AIH, but take it away.
Yeah, so that's a big question, and I think it's as Naheed again leading the answer is something that we are thinking about, both at Valo, but also more importantly at the Alliance for AI and Healthcare. One of the things that we want to be doing is bringing together these data sets. We know these data sets exist. We know cleaning them is a large pain. And what we don't want to do as an industry is to have every company, say we've even made it available. There's availability. First problem is availability. Second problem is cleaning. And if we make it available, every company is going to run through the same process. We're going to waste, I don't even want to fathom the number of man hours that we end up duplicating effort as an industry, which is not helping patients. It's just getting the data into a form that can be used. So, one of the things we're doing at the Alliance for AI in Healthcare is we have a number of data initiatives, and we're interacting with organizations that have these data, and trying to figure out how we can bring them forward, how we can put together a common strategy, at least to clean up, do the first initial steps of normalization, regularization, and cleanup, that make it a bit without making any assumptions, right? Because everybody's use of the data is going to be slightly different, and so we don't want to impose any assumptions on that data in terms of transforming it. But unfortunately, I would say, there's the efforts of the Alliance for AI in Healthcare, which I just talked about. There's also more recently an organization that's emerged called the Data Alliance, and again, they're working specifically on almost more specifically on this exact problem, and they have a number of efforts that are happening. We are interacting as the Alliance for AI in Healthcare with them and making sure that we bring our efforts together. So I'd say, and also more recently, what's interesting, so the White House, or OSTP, the Office of Science and Technology Policy, reached out to a number of us. I'm sure our other panelists were probably invited as well, but I was in one of those sessions when they were talking about ARPA-H. This is the new initiative from coming out of, backed by the White House and others within Congress, to essentially apply a ARPA type approach to healthcare. And one of the reasons the White House was convening these panels was to understand, well, what are the big problems that they should be focusing on? And I can't tell you, like, for an hour, they just got pounded with, every time they asked the question, it was like data, high quality data. Like if you're going to invest, if you're going to spend money in this industry, spend money on making it, bringing data sets together, making them more widely available. So I don't know any specific, maybe our other panelists have them. I don't know any specific efforts out, at least within the United States. Like I said, recently the Maccabi has more publicly announced that they're working with individuals. So they've done a very good job of organizing this Israeli data within their system. But I think there's becoming more and more pressure on the United States and the European Union to produce these data sets. And I expect that in the next few years, more of those efforts will come to fruition, and you'll see it. But right now, I'm skeptically optimistic. Maybe my other panelists already know efforts. I know there are efforts in the UK as well. Sorry, Andreas, go ahead.
Oh yeah, sorry. When there's data, there's availability, right, and pooling data, and that's what you described and what Julie asked for, right? How can we annotate that as XML, as some tagged format or so, right? So it can be used in a standardized form. What you sometimes see also in consortia is that, especially in the preclinical area, for example, here in Europe, is that data gets just pooled that has been generated for completely different purposes years ago, but where there's no experimental design, so there's no sampling of chemical space in any way. It's probably compounds that are not particularly interesting. There's no controlled vocabularies or so that are used to annotate the compounds, and that's then actually quite difficult to put into any kind of meaningful models. There's no proper sampling of chemical space, of mode of action space, and also not of endpoint space. So it's very difficult to use that for models. And I want to come back here to Ali's points, finding patterns in data. In chemical space, that can be quite tricky. I remember when I was still a PhD student, we were looking for features in chemical data sets that make compounds bitter. What did we find? It's sugar rings. Why is that? Because the confounding factor, right? Quite often natural products are bitter, and quite often they are glycosylated, right? So you can have huge confounding factors there. And in chemical data sets in particular, quite often you have chemical series, right? You have analog compounds and so on, It's very tricky actually to find meaningful patterns in chemical data, just as a word of caution,
Thanks, Brandon. Thanks, Andreas. And I think that shifts nicely into the next set of questions and thoughts that I'd like to extract from this group. So let's jump into outcomes from AI models and their relevance to specific problems, and in particular into drug discovery. So there's a lot of effort and investment going into the discovery of better medicines using multiple types of approaches. You've heard about AI drug discovery, iPSC human organoids or tumoroids. A disproportionate amount of effort and investment historically, and I think still probably today, is in small molecules. But not every molecule can be turned into a drug. I've personally heard feedback from chemists in pharma who look at outputs from other AI models and cringe at some of the molecules. I once witnessed a chemist throw a tantrum while an AI presenter showed some molecules on a presentation. Felt very bad for that presenter. So let's talk about using AI to identify viable drug-like molecules. What goes into that? Pat.
Yeah, I think a lot of this is a matter of mindset. And I think we have to get away from the way that we've typically done drug discovery. For years, we did experiments the same way that we always did, and then handed the results of those experiments to a computational group and said, here, make a model from this. And the computational folks would look at it and say, but this is not the data that we need. This kind of comes back to Andreas's earlier point about, you've got just highly congeneric series, or you've got a set of molecules that are irrelevant. And I think what you have to do is integrate the building of models into your process and really start looking at this from the standpoint of which experiments can I do to help me to build the best model. And I think if you get this integrated mindset between experiment and computation, you'll make progress. I don't know that you'll make progress by sticking with the standard way of we're going to run drug discovery the same way that we always did. And here, take this data and build a model.
So I'm going to build off that for a second. There's a question that came through the chat, and the question reads, what would you say is the most challenging I'm going to guess there's a missing word, aspect in applying AI ML for drug discovery? Would you say it's that?
Yeah, it's making sure, I think, this comes back to what we were saying earlier. It's all about the data, and it's generating the right data, and it's doing the right experiments, and it's having this tight integration between computation and experiment. To me, that's the most critical piece.
Yeah.
So I know, Brandon, you have a comment.
Yeah.
I just want you to touch on, as you go through this, on chemical space exploration, synthesizability, stability, getting a molecule that can be scaled up into large batch synthesis as well.
Yeah. So again, but I go back to what Pat was saying, and it's absolutely critical that data science is not seen as a service for therapeutics, right? That therapeutics and data science, honestly, I hate the term data science because all science is data. And at some point, I hope we get rid of that term. No offense to the data scientists in the room, but science is data and data science is science. But the thing is that right now there's this, especially in pharma, this kind of hierarchy, and data science is often in service. And what we do, and I know Pat at Relay, what they do is spend a lot of time making sure that it's a partnership, that people are making decisions, that data scientists have influence on what experiments to do, when, what data to generate, and really define a data-driven process. I think one of the aspects of often that gets brought up, however, and this is what Naheed alluded to, right, is that a data scientist presenting something and a medicinal chemist having a tantrum over some aspect of unsynthesizability. One of the things that we think about, or the way that I think about it, is often things like synthesizability and other things in the initial phases are overemphasized, right? In that, in the beginning, the way we think about drug discovery is not about, right? This is not no molecule left behind, right? That's the old way of thinking about it. No molecule left behind, right? The problem is often the way that we think about it in the early stages traditionally is that, oh, this molecule might be the one, or this series might be the one. And so we have to make every single one in this series. But really what we should be thinking about is what's the data content in synthesizing that molecule, testing it, and adding that data back to the model, because odds are infinite that you are not going to, in your first round of synthesis, even your hit finding, you're not going to find the drug. So what you should be asking is every time you spend money to synthesize or do something, is think, what's the data content? Is this, this one has a schmethyl and this one has an ethyl. Why make both? Let's just make the one with the schmethyl. And because it's really about the data that it's going to bring back to the model that's going to then help us ultimately find the drug. So that's in the beginning, but then in the later stages, one of the things I often see is then synthesizability and becomes not important enough. And when I mean by scale up synthesis, right? One of the things that I've seen kill programs over and over again is no one thought about big batch synthesis. No one thought about how can we produce enough of this drug to actually make it a drug to put into humans, because the synthetic routes you're going to use to do small batch 5 grams is totally different than when you're making gallons, making liters, and you're making these pounds of this stuff. And so one of the things we need to think about, and one of the exciting things is can we build models of synthetic scalability and CMC, and think about that in the molecule design phase when you're sitting there thinking, okay, I've got candidate 1, candidate 2, which one do I push forward? Actually thinking about CMC is one of those things which is often not thought about until we're already in the clinic, and someone in CMC is freaking out because they don't know they can't figure out how to up synthesize these things. So I think there's kind of a double-edged sword. I think we worry about it too much in the beginning, like the story that Naheed gave, but we also often think about it too little late when we think about full scale up.
Any other comments from the panelists on that? I do want to give a shout out to the question that Julie asked. There's one company out of Paris by the name of Auken, Thomas Clozel, building a federated data learning platform to aggregate a bunch of clinical data without having to share that data and have models be accessible to companies. I think that's one really unique way, and I like what Auken's doing really well. And then there's a company, I'm not overly familiar and expert in kind of the scale up space nearly as much, but Crystalphi out of Asia, big company, raised a lot of capital. I think they popularized themselves kind of in polymorphism and scaling up in the chemistry and in preclinical development. A really interesting problem that I don't think nearly gets enough attention. I want to stick on this topic for a second, and I recognize we only have 9 minutes left, but there's a really nice question that came through that I think we have, Pat, as your expertise from your current company. Question is, what challenges do you face, or does the industry face, with data in signal to noise ratio in ML applications for areas such as DNA encoded libraries, DEL systems?
Yeah, and a lot of that comes back to having this tight connection between computation and experiment. So if you're going to build a machine learning model based on DNA encoded library data, you need to have a very tight connection between the group building the DNA encoded library, the group doing the sequencing, and the group building the machine learning models. So you really have to go through and do the appropriate experiments and say, okay, what depth of sequencing do I need in order to build a viable model? So I think it's really, all of this comes back to feedback, close collaboration between computation and experiment, and making sure that you're generating the data that's the best data to build a machine learning model. Because if you're doing DNA encoded libraries, the data quality that you need, if you're just going to do off DNA synthesis, may be different from the data quality requirements for building a machine learning model. So, I'm just going to keep saying integration of computation and experiment. You're going to get sick of hearing it.
It's the most important thing to accentuate. And for those of you wondering why I turned to Pat with that question, His company, Relay Therapeutics, acquired Zebi AI, a great company that was in the Dell space, that is in the Dell space. So I think it's a really interesting combination of technologies, but goes to AI is not the silver bullet. You need to combine different forces, different technologies, different methods, different ways of thinking to get to that endpoint faster. So I want to shift gears to kind of the last question, the set of questions, and this is on machine learning systems as a life cycle. So considering all those necessary considerations and feedback regarding chemical viability, synthesizability, stability, etc., that Brandon, Pat, and others just walked through, how do you think the technology needs to be set up to incorporate this knowledge continuously? And for this, I'm going to turn to Ali and Andreas.
Thanks, Naheed. And I want to add the cautionary comment that Andreas said about reliability of the patterns, for example. And that is very important because we need to consider that machine learning modeling at industrial level is not a one-time task. So it's a life cycle. So we have to go through this continuous improvement. We have to be able to collect feedback and incorporate that feedback from different kinds of expertise that we have in the company, from medicinal chemists, if they can add knowledge and improve our models, from even from machine learning perspective to assess explainability of our model, reliability of the patterns we are identifying. So this is what we've done at Cyclica. So we try to come up with a scalable system to be able to incorporate the feedback and be able to benefit from different modeling. It could be in the representation learning and featureization process. It could be about assessing reliability of the patterns and predictions, because as I said, it's not a matter of building a machine learning model, getting a high ROC AUC, PR AUC, and then putting it in production. That's not how it works. So, and having this pipeline that goes through this continuous improvement, collect the feedback, add new data with proper assessment of reliability and quality and relevance of the data matters a lot. So there are some, for example, in some aspect of it, some best ML ops and DevOps practices that can help for reproducibility of the results, to do model versioning and data versioning. There are explainability best practices to make sure about reliability of the predictions. There are tools out there that can help us and could actually have been helpful for us at Cyclica to incorporate the feedbacks without the necessity of expending weeks and trying to manually change some of the codes or some of the processes that we have. And that's something very important when we are building a technology we want to use for months and years.
Yeah, absolutely. And that whole ML ops area, right, that is still moving very fast. It's quite fragmented, right? It's a painful area to deal with. And that's actually an area where we discussed together with Cyclica as well, if there's any way to form some type of open source consortium, right, in order to move our field forward. And we still need to figure out the details on that. We had some chats on that. And if there's any feedback from the public, feel free to get in touch afterwards. So very happy to involve the audience here as well in that.
I'd love to join that. I didn't know about that. Like, I spend an inordinate amount of time thinking about ML ops and thinking about it at scale. It's still, you know, we just recently at Valo looked at, I think, between 20 and 30 different solutions out there for thinking about around ML ops. There's a lot of really good companies. A lot of things have moved in a very positive way, but obviously in thinking about translating the interesting thing that is unique to, well, not unique, but somewhat unique to this industry is just the number of different modalities of data we're dealing with. Genomic data, image data, longitudinal patient data, chemical data, biology data, like all of these different types of data. And to think about ML ops in their ability to build the model, you know, building a model on genomics, building a model on imaging, being able to handle all of those, because most of the time what you find in ML ops is, you know, the problems that companies are dealing with are much narrower, and so they build a more bespoke and specific ML ops platform and pipelining system for that, but we're dealing with so many different types of data, and not only that, active learning is a huge part of it, as we've said many times, right? And so we need to be able to iterate and build those cycles quickly, and with all of these different types of data sets. So sign me up, because I think about this. This occupies, unfortunately, because this is kind of the plumbing, but it occupies a fair amount of what I think about on a daily basis.
Yeah, will do.
Excellent. So with the 2 minutes we have left, I want to thank the audience for being here today. I really appreciate you spending the past 58 minutes with us. And I'm going to go off the beaten track a little bit and challenge the panelists with 30 seconds each. And I'm going to go Pat, Andreas, Ali, Brandon, with the following question. 2030, 8 years from now, we talk a lot about AI and drug discovery today. What will be the topic of conversation as it relates to our space? Will this be the conversation, or will we get beyond this? And what does the world look like in 2030 with technologies, some of which we're spearheading today? Pat.
Yeah, I think this is going to come back to the very beginning of our discussion. And in 2030, we're going to be talking about how do we do the most physically relevant experiments for patients? How do we collect that data in a robust fashion? And how do we use that then to build models that enable us to get to therapeutics?
Andreas.
Yeah, what I very much hope that we move towards until 2030 is consortia involving experimental design in order to sample a hypothesis space properly. So not just sharing data that is there already, but consortia that designs data experimentally, right, to generate the right data that we actually need to build in vivo relevant models. That's what I really wish for.
Brandon.
Yeah, I would say by 2030 we will be having discussions with the FDA around which is the next set of animal models to get rid of, right? Because we're actually able to now, we've shown them enough times that the machine learning models are good at predicting how compounds are going to interact in humans, to the point where a lot of animal models will disappear. And so the discussion at that point will be, what's the next animal model to get rid of, and how do we also think about clinical design? Can we get rid of a phase 1 at that point? Again, we'll be having those interesting discussions. I'm looking forward to those.
It seems like Congress in the US is also pushing that, and one of our friends and partners, Meredith Brown-Tuttle, VP of Regulatory Affairs at Recursion, recently said at Bio International on a panel in June, where the day of going target to clinic without touching an animal is in front of us and not too far in front of us. Ali.
Yeah, so as Pat said, so this is a starting point of putting physics-based and chemistry-based and ML-based kind of techniques together, and I'm hopeful that actually it's going to get better and better and become standard in the industry. But something I'm very hopeful for, which is in line with what everybody said, is bridging the gap for translatability from preclinical drug discovery and bring it more successful drugs into clinical setting. And it's not only a matter of more drugs, it's actually a matter of less expensive drugs.
And with that, Pat's on his move to his next meeting. So I'll finish off with 3 things. Number 1, balanced data, heterogeneous data, so that we can train based on clinically diverse. Number 2, mindset. And AI ain't the silver bullet. Human intelligence plus AI plus other technologies is critically important. And number 3, collaboration. No one company is going to do it itself. So with that, to thank you to everyone. I appreciate the panelists making their time, and hope to see you all the next M2M series next week. Cheers. Bye.